Unit 11, Lesson 5, Genetic Diseases and Gene Technology. So I wanted to recap this just because I noticed some people are still struggling with this concept. So homo means same. In an easy way that I came across this and I was like, I want to share this. Homozygous. So like you could think of it like they're homies. They have the same thing. They like each other. They do the same thing. They're homies. Homozygous. So that means like that would be both big A or it could be both little a, right? They're the same capitalization. They're the same trait. Boom. Heterozygous are going to be very different, right? Hetero means different. So that's going to be big A and little a. Okay, homozygous, heterozygous. So think of it like a homie. Maybe that'll help it stick in your brain. All right, lesson objective is that students should be able to identify the different uses of genetic technology, and we will explore different genetic diseases such as Down syndrome. First things first, we have a warm-up question. So in humans, your blood type is determined by the A, B, and the O alleles. You're going to set up a test cross for a parent with the genotype and a parent with the genotype AB. Sorry, I went and I changed what I'm doing for the warm-up just because I noticed last time like people didn't know what they were doing because they were just copying what I said. Okay, so genotype. So my example is going to be different than the one that's on your worksheet. So we're going to talk about genotype B and genotype AO. So I can show you guys what to do, and then you will do your own warm-up on your own. Okay? First things first, we have to look at the parent's genotypes. Genotype B, genotype AO. We're going to put it on this Punnett square and move on. So remember that allele A is going to produce antigen A. Allele B is going to produce antigen B, and allele I produces no antigen. So for blood type A, we could have IA, or we could have IAI. For phenotype, for blood type AB, IAIB, so forth. Okay, if that doesn't make sense to you, pause the video, look at this chart, and try and figure it out. If you're still confused, raise your hand. All right, so we have a parent with genotype B, so I have IB. Parent type with genotype AO, IA for antigen A and I. Okay, so for yours, it's a little bit different. Let's keep moving. Karyotypes. So karyotypes are going to be a fancy picture of all the chromosomes in a specific organism. These chromosomes are found in the nucleus of every cell in the body. So that's when they take every chromosome, they lay them out real pretty and nice, they stain them, they make them look cool, and they look at them under fancy imaging software. Karyotypes can be used to diagnose diseases. So we know what a normal karyotype looks like. We know the normal size of each chromosome. We know the normal shape of each chromosome, and we know the normal number of chromosomes. So by looking at a karyotype of someone who we suspect has a genetic disorder, we can confirm or deny that they have the disorder by looking at their size, shape, and number of chromosomes. So for example, this syndrome called Edwards syndrome, all of the chromosomes are normal except for the ones at place 18. The ones at place 18, normally we're supposed to have two, right? We get one chromosome from mom and one chromosome from dad. But because of a non-disjunction event occurring in meiosis, they ended up with an extra chromosome in there just because they failed to separate fully. That happens sometimes. This person has Edwards syndrome. Then we have what's known as Patau syndrome, which is an extra chromosome at place 13, and Klinefelter syndrome, which is an extra chromosome, an extra X chromosome. You don't have to memorize any of these syndromes. I'm just telling you what they are. Of course, there's this famous syndrome known as Down syndrome, which is aka trisomy 21. What does trisomy 21 mean? Well, tri means 3. Somy means chromosome, so 3 chromosomes at place 21. Boom, boom, boom. 3 chromosomes at place 21. That gives people certain characteristics that are Down syndrome characteristics. Pedigrees. Let's move on from karyotypes into pedigrees. So pedigrees are charts that show the presence or absence of a certain trait within a family across different generations. So you may have seen something like this before, where you show the presence or absence of a trait. It could be a disease, such as hemophilia, cystic fibrosis, Huntington's, or it could just be a random trait, such as purple hair. Okay, let's see how to read a pedigree. It's pretty straightforward, so just stick with me. So first things first, squares are going to be representing males. Circles are going to represent females. So all the squares are males and all of the circles are females. All of the shapes that are filled in are going to have the trait or the disease. All the shapes that are empty do not have the trait that we're looking at. And sometimes in some pedigrees you'll see these things are half and half. That just says that this person is a carrier. That means that they do not have the disease themselves, but that they do have the potential to pass it on to their offspring. And of course, we have generations. So this is the first generation of this family, the second generation of this family, the third generation of this family. So let's do some practice problems. Make sure that we understand this concept. So how many males are there in this family? Well, let's count them. 1, 2, 3, 4, 5, 6, and 7. So there are 7 squares, there are 7 males in this family. How many affected women are there in the third generation? Well, let's look at the third generation. So we're looking at these organisms. How many women are affected? Not that one. This one's affected, so we have 1. Is that one affected? No. So we only have the 1. It's only 1 affected woman in the third generation. Genetic engineering.
So, what is genetic engineering? Genetic engineering is when the genetic code of an organism is altered to produce a desired trait. This is done by taking a gene from one and inserting it into the DNA of another to get the wanted end result. To get a better sense of how this is done, let's first understand how DNA works. In any living thing, the DNA acts as the instruction manual for the body. Certain genes code for certain proteins, which in turn make up all the different parts of the organism. Any trait that you can think of is coded by DNA. Now, once you know what codes for what, you can use genetic engineering to essentially edit the parts you want, to some extent. All living things use the same genetic code, DNA, made from the same 4 bases, so scientists can use special enzymes to cut parts of the DNA from one living thing and sort of sew them onto the DNA of the target. The result is then a living thing that has a new gene and a new feature as a result of that gene. Now, there are many examples where genetic engineering is used today, but we're going to focus on a very specific medical example to understand this concept better. Humulin, a medication used for diabetics, is created with genetically modified bacteria. Humulin can be given to diabetic patients who lack the insulin they need. Now, how they do this is scientists figured out which section of our DNA codes for insulin, and they also found certain enzymes that can cut that gene out. What they do is, using enzymes and heat, they isolate specific gene and then insert it into the DNA of bacteria. The result is now a bacteria that produces human insulin. And since bacteria can multiply really fast, you can create a lot of insulin in a very short amount of time. Now, this is great. Genetic engineering poses huge potential in the field of science, providing us with new insight on how our genes work. However, there's also a lot of controversy in certain respects to genetic modification, specifically genetically modifying our food. To learn more about GMO foods, check out our next video.
So now that you've learned about that, let's look at who owns genes. So do you think the company should be allowed to own parts of the human body? This is an ethical debate beginning in the 1970s when companies and big research universities began seeking patents on genes. Private companies began researching human DNA and genetic diseases so that they could patent them and charge other people money for researching and helping to cure those same diseases. What is a patent? Well, a patent grants exclusive right to make a profit from the sale of an invention. So if companies own patents on genes, then they can refuse to allow other scientists to use those genes in their work and their research. This extra limitation could potentially prevent important life-saving discoveries from happening. Also, getting gene therapy or gene testing is so expensive if the gene is patented. Medical debt is already ridiculously crushing for many Americans, so an extra barrier due to costs would no doubt lead to very preventable death and suffering. So most of you guys might sit here and be like, well, I love capitalism. I want, if they were the ones who looked it up, then they should be the ones to get all the money for it. Well, think about this. There's an alternative. So this guy named Jonas Salk, he developed the polio vaccine. You might think, what's polio? I was born in 2004. I don't know what polio is. Well, basically our grandparents had this major disease that was just part of their daily life, known as polio. It would get into your leg bones and it would basically mess it up to where you couldn't walk anymore. And obviously not being able to walk affects people's lives drastically. So polio was paralyzing between 13,000 and 20,000 people every year. There are 365 days in a year. 20,000 people each year were getting polio and being paralyzed from it. So Jonas Salk could have made lots of money off of this vaccine, but he decided not to patent it. He said, no, everyone should be able to get this vaccine. Let's get rid of polio virus, get everyone vaccinated so that we all can walk and live and be happy and healthy. So, people thought this man was crazy because he could have been a billionaire, but he decided not to. He decided that he would give it away to people, and now we have people who are 15 years old and they're like, what's polio? I don't even know what that is. And that's because of this dude right here, Jonas Salk, an American hero. Everyone say, thank you, Jonas Salk. Cool. Farmers deal with it too. So this isn't just a thing about medicine. This is also people who grow our food. So agricultural is a multi-billion-dollar business. Billion with a B, as in take 1,000,000, times that by 1,000, and then you have 1 billion. Then take that and multiply that. Okay, so just realize there's lots of money in this business. So some companies decided to patent their specific strains, their specific GMOs, so that only they can grow their variety of the crop. But the problem is, wind and rain exist. So wind and rain may take seeds from the company's fields and blow them onto someone else's fields. If the company finds out that these seeds somehow blew onto someone else's field, the company actually has legal standing to sue that farmer. And as we know, courts often rule in favor of large corporations because they have the money, they have the time, they're able to go and fight these long, huge court battles. They also can spend money on ads and spend money on all kinds of other things to try and sway public opinion in favor of the corporations. If you're interested in stuff like that, if you're interested in seeing how corporations flex their muscle on everyday people, look up McDonald's hot coffee. Just look up McDonald's hot coffee and you will see exactly what I'm talking about. Even though it's impossible to stop nature from spreading seeds, courts don't care. Companies don't care. They will sue whoever it is. So, knowing what you know now, what is your opinion? Should corporations be allowed to patent genes just because they might have been the first to research them? Or should genes remain property of all of humanity and all of life on Earth? What do you think? Well, here's what the Supreme Court thinks. In June 2013, the U.S. Supreme Court unanimously ruled that naturally occurring genes and genetic sequences are not patentable. Basically, they invalidated patents on these breast cancer mutation genes, and that made it so that all other gene patents were also invalidated. So luckily, the Supreme Court ruled in favor of the little guy, less regular folk. However, we also know that currently there are lots of battles over genes and over gene technology and whether or not it's ethical and all of that sort of stuff, especially when you start to bring in like Ancestry.com or like cloning and stuff like that. So if you're interested in this, I would just look up gene technologies and see what kinds of crazy cases there are out there, because there are a lot of cases. This is a very widespread thing. So yeah, cool. Gene technology.